we'll be talking about bacteria and more precisely lactic acid bacteria in wine. Forgot to mention that there'll be a Q&A session at the end of the presentation, but feel free to post your questions whenever you want during the presentation. Okay, so the way the presentation is going to go is that we're going to do a brief overview of lactic acid bacteria in general, and then focus more on Inococcus ini and its malic acid metabolism. Then we'll go over the citric acid metabolism in Inococcus ini that is directly linked to diacetyl production. And we'll finish with a brief overview of different malolactic starters. During this presentation, I will use some abbreviations, LAB as for lactic acid bacteria, MLF for malolactic fermentation, and written but not said, ONI for Inococcus ini. So you may not know that because they are smaller than cows, but actually lactic acid bacteria is one of our favorite domesticated living organisms. We can find it in, and it is used, they are used in many dairy products such as yogurt, cheeses, but are also used to produce bread. They're used to ferment meat products such as sausages, and they are also very useful in fermenting vegetables such as cabbage to produce sauerkraut, but also fermenting cacao, tea, etc., etc. Many different species are involved in this fermentation process. I've listed here a few, but it's not at all an exhaustive list. And of course, one of our favorite domesticated animal, at least in wine, is Inococcus ini, and we'll talk about it more deeply a little later. The main roles of lactic acid bacteria in general in a food perspective is to acidify, thus stabilizing the different products they are fermenting. But they are also involved in aroma production because beyond the acidification, there are different aroma that are produced through their fermentation pathways. And they also have some probiotic properties, but I will not go over this more. So there's one thing that all lactic acid bacteria have in common is the ability to transform sugars and in dairy products, it's lactose, into smaller sugars and then lactate. This is where the main acidification pathway is involved. But if all of these lactic acid bacteria do have this common feature, the different species that the group or consist this group do evolve in very different ecosystems. As a comparison, you can see that in milk, the pH is usually close to being neutral, while in wine, it's very acidic. In milk, there's no ethanol. Well, there's some ethanol up to 15, 16, 17% in wine. And wine is very poor in nutrients, unlike milk, and it also has very little sugars and some inhibiting compounds. So that's why we do find many different lactic acid bacteria that are fitted to specific environments. And you can see here a phylogenetic tree showing you a few of these different species. And for example, some lactobacillus are more involved into producing yogurt, as well as streptococcus, while Inococcus ini is particularly interesting for us in the wine industry. Here is a graph showing you the evolution of the different lactic acid bacteria species that you can find before and after alcoholic fermentation. And you can see that in must, hence before alcoholic fermentation, you do find many different species, such as some bacillus, but also some lactobacillus, such as Lactobacillus plantarum or Leuconostoc mesenteroides. Inococcus ini is just a part of this lactic acid bacteria ecosystem at that point. However, when must is fermented by yeast and gives wine, then Inococcus ini is one of the only species that is found, at least considering lactic acid bacteria, because it is particularly adapted to wine, because it's resistant to ethanol and can stand low pHs. So really, Inococcus ini has really very specific abilities that makes it one of the most fitted lactic acid bacteria to stand the wine conditions. Inococcus ini can be differentiated from other lactic acid bacteria that are present in the wine-making environment. Again, I said that Inococcus ini was one of the most fitted microorganisms for wine, but you can still find some different species sometimes, such as Pediococcus or Lactobacillus, especially Pediococcus damnosus, Pediococcus parvulus, or Lactobacillus plantarum, brevis, or Elgardio. All of these latest species that I've mentioned, you can usually find in must, but rarely in a wine environment with ethanol. Rarely you find them, especially when the pH is high. Well, Inococcus ini are cocci that will form long chains, and they're egg-shaped cells, whereas Pediococcus are also coccis, but they form tetrads, so they're very different from Inococcus ini. They have a very different organization, so that one can easily distinguish them under the microscope, and more easily can one distinguish Lactobacillus, which has rods and that usually form long chains as well. Another feature that differentiates these lactic acid bacteria is the way they behave by metabolizing sugars. Inococcus ini, when degrading sugars and moreover glucose, fructose, and some pentose, will degrade them in lactic acid, but not only. It will also produce from sugars ethanol, acetic acid, and CO2, whereas Pediococcus will only produce lactic acid. Lactobacillus may form lactic acid, and sometimes, depending on the species considered within the gender Lactobacillus, will form sometimes ethanol, acetic acid, and CO2 as well, but not always. Again, it depends on the species you consider within the Lactobacillus gender. One of the main roles of Inococcus ini in wine is to carry out malolactic fermentation. I talked about sugars, but usually in wine there's no longer sugars, and this is not, sugar degradation is not an activity the winemakers look for, considering Inococcus ini in wine. Its main role is to carry out malolactic fermentation, which consists in the decarboxylation of malic acid into lactic acid, hence bringing a deacidification and also microbial stability, because it will remove substrate, the first one being, of course, malic acid. And it will also participate through secondary metabolisms to aromatic modulation. Malolactic fermentation can occur spontaneously in wine with indigenous strains. Sometimes it can be problematic. That's why the use of selected starters has been developed. And nowadays, most of the malolactic starters contain one species, which is Inococcus ina. You can sometimes find some starters composed of Lactobacillus plantarum, but very rarely. And usually it's one species and one strain that is isolated, identified, and selected in laboratory, and then that is produced at an industrial scale and then inoculated in wine. The reason why people use selected starters is because their selection through a specific strain makes it a robust product to use, whereas indigenous strain can be a little more difficult to handle. They are also selected so that they're unable to produce biogenic amines or other off products. And most of the time, the malolactic fermentation duration is low, is shorter. Actually, the story of the use of Inococcus ina as starters in wine is not so old. It was only in the 60s where Pinot and Domergue started to describe the use of fresh bacteria involved in malolactic fermentation. But that was actually this first description, a use of lactic acid bacteria mixed with yeast in must. And we had to wait 10, 15 more years when the first description of a pure fresh starter, again in must, was described, and that was in 1977 and 1980. And at that time, the species that was the name of the species that was used was Leuconostoc unos. The taxonomy of Inococcus ina went through a lot of different terms, going from Bacillus gracile to Leuconostoc oinos to Leuconostoc unos. That was a formal first description of the species responsible for malolactic fermentation in wine. And in 1995, it was reclassified as a new species, Inococcus ina, because of the development of DNA analytical tools that enable taxonomists to show that it was really a genus apart from the Leuconostoc group. Nevertheless, research continued to use Inococcus ina, or what was still called Leuconostoc unos, as a starter in wine. And LAFFORT and collaborator in 1983 described a first frozen starter in wine, but that needed to be acclimatized first. All of the other research before, again, had focused on inoculation, but in must. So acclimatation was needed to inoculate it in wine. In 1996 was described the first direct inoculation in wine with no more acclimatation needed. And nowadays, I haven't done the exact count, but more than 40 commercial strains exist on the market. The evolution of the selection of Inococcus ina as a starter in wine has evolved with evolving knowledge on this species and evolving tools to be able to select starters on more and more precise criteria that for a long time relied on phenotypic criteria and nowadays rely on more global genome analysis, but I will not go into that deeper. We can talk about it in the Q&A session if you want. Coming back to the general metabolism of Enococcus ani, in general, just like any other lactic acid bacteria, Enococcus ani can get some energy to develop and survive through sugar metabolism. And the way it does it, and I'm not talking in a winemaking condition, I'm taking in general conditions, it will use glucose or pentose to produce ATP that you see here, that is the molecule that has a high energy containing bond, that is the energy reserve of the cell. The problem is that in a winemaking environment, there are no sugar left to ferment, and Enococcus ani needs to find other ways to gain ATP, to gain energy, to be able to multiply and maintain itself in wine. How does it do that? Well, malic acid can be an energy source for Enococcus ani in wine. Pentose and citric acid can also be energetic sources, but malic acid is one of the main energy sources in wine, but it's an indirect energy source. And let me take a few minutes to explain that a little more in detail to you. Malic will enter the cell in an anionic form and will immediately, as it goes into the cell, consume a proton to produce lactate. This proton consumption enables the cell to maintain a pH gradient in between the outside of the cell and the inside of the cell, enabling proton to be able to go in the cell through its concentration gradient through what we call an ATPase, this which is actually a proton transporter, that as protons go through this transporter will be able to synthesize ATP. Okay. So indirectly, by maintaining what we call a proton motive force, cells will gain energy through ATP synthesis. Okay, just because the malate will be able to consume a proton through the malolactic fermentation. In wine, there are variable concentrations of malic acid, roughly, very roughly, ranging from 0.5 to close to 9 grams per liter throughout worldwide wine. But it is true that lower and lower concentrations are found in must in wine because of climate change and also later harvesting times that are being processed. And nowadays, it's not unusual to have to perform malolactic fermentations in wines where malic acid concentrations are around 1 grams per liter or even lower. And lately, we wanted to investigate a little deeper what were the consequences of these different initial malic acid concentration in wine. If malic acid is an energy provider for Enococcus ani, what does that mean in terms of malolactic fermentation performance in wines with different concentration of malic acid being in wines? So let me show you these results in detail. What we found out is that variable malic acid concentration did lead to variable time to achieve malolactic fermentation. And the way we did that is that we spiked a wine that had a low initial malic acid concentration with increasing malic acid dose. Therefore, having wines ranging from 0.6 to 6.3 grams per liter initial malic acid concentration. Of course, because by doing that, we changed the pH of the wine. We adjusted the pH of these wines so that the only parameter that actually changed it in the wine was the malic acid initial concentration. And you see that the higher the malic acid, and we did that in 2 wines. I'm showing you this with one strain that is called B7 Direct, but we actually did it with many different strains and starters, and the results were always the same. So the higher the malic acid concentration, the longer the malolactic fermentation takes to achieve. But for very low concentration in malic acid, they seem to take a little long than for concentrations around 2 grams per liter. And to illustrate that, we calculated the malic acid consumption rate, meaning the malic acid that were consumed in milligrams per liter and per day. And you can see that in both wines, one wine that we called red wine, the other wine was a more precisely a Merlot, that the lower the initial malic acid concentration, the lower the mean MLF rate is. So weak malic acid concentration do reduce the fermentation rates, but they do not by all means prevent malolactic fermentation from being carried out. So it's not a limiting factor. Okay. We wanted to know a little more about these low rates when low malic acid concentrations were present in wine. So what we did is that in one of these wines, we perform simultaneously 2 malolactic malic acid monitoring, population evolution monitoring according to malic acid concentration. And this is what this graph shows you. Here you have a same wine. The only thing changing is the initial malic acid concentration. In one situation, it's close to 0.6 grams per liter, and you have here the malic acid evolution throughout time. And in another modality, the initial malic acid is at 3.5. And again, it's exact same wine, except that it has been for one assay spiked with malic acid. And you see that the time to achieve malolactic fermentation is longer when higher initial malic acid concentration are present. But what you also see is that for this higher initial malic acid concentration, you have very high population developing from a same initial population close to 5.5 10 to the 5th cells per mil. They almost reach 10 to the 7th cells per mil as a maximum population, whereas when you have an initial concentration close to 0.6 grams per liter of malic acid, the maximum population barely touches 10 to the 6th cells per mil So there's a smaller maximum population with a lower initial malic acid. And we continued by calculating the maximal rate per cell. We already had the mean rate, and again showing that the rate is lower when you have a lower initial malic acid concentration than when you have a higher malic acid concentration. But if you bring this back to what happens to a single cell level, and we could do that because we knew the cell levels during malolactic fermentation, you see that each cell actually metabolizes very similar malic acid contents throughout malolactic fermentation, and even it seems like they're able to metabolize a little more malic acid with lower concentration. What does that mean? That means that there is a substrate limitation with lower malic acid concentration, limiting the maximal Inococcus eni population in this situation, leading to a smaller global malolactic fermentation rate, but a same or even higher individual cell rate. Okay. Again, this means that malic acid concentration doesn't impact the cell performance, but less cells are present, so it will impact the overall general malolactic fermentation rate, even if it will not prevent malolactic fermentation to be carrying out. I hope this is clear. Please, if it's not, go on with the questions, and we'll go through it again in the Q&A session. So if we try to translate this, what we learn, into practice, let me remind you that malic acid is an indirect energy provider for Inococcus eni in wine, and that when malic acid is present at low initial concentration, the bug development is limited. Again, it doesn't prevent malolactic fermentation from being carried out, but you can have other limiting factors that are present, such as high ethanol, low pH, or other unknown parameters. And I think we all have been confronted to situations where malolactic fermentation is very hard to achieve, and we do not understand why. And in this case, it might be useful to boost this population, for example, with a higher inoculation dose. And let me explain that a little further. The minimum population to start malolactic fermentation is 10 to the 6 cells per mil. Okay. This is, for example, the population you have when you inoculate with a starter. But you always have a little drop. And let me go back to this slide here. You see, you can inoculate at or close to 10 to the 6 cells per mil, but you will always have a small population drop that can be exaggerated if you have important inhibiting factors present. So if this drop is too important, it's going to be hard for the population to come back to its initial population of 10 to the 6 cells per mil, meaning that it might be helpful to inoculate at a higher dose. Usually the dose that one inoculate with is 1 gram per hectoliter, and you see it in this situation here. And when we inoculated in this wine, which was fairly hard to achieve malolactic fermentation, it took more than 30 days. Double dose really helped to reduce the time of malolactic fermentation, because the time to recover this 10 to the 6 cells per mil was shorter. So I'm done talking about malolactic fermentation, but remember, Malolactic is an indirect energy provider for Inococcus ini. It's one of the main energy provider because in wine there is no sugar. There's another substrate that can be used as, again, an indirect energy provider, and this is citric acid. I'm not going to go into details, but likewise, citric acid will indirectly lead to ATP synthesis through ATPase functioning by proton entering into the cell. Citric acid metabolism will, on one hand, lead to indirect ATP synthesis, on another, directly lead to diacetyl formation, but not only. This link here is direct, but it's much more complex, and we'll see that in the next slides. For the winemaker, the consequence is an organoleptic modulation through citric acid metabolism, because diacetyl is a molecule that is able to bring buttery notes to the wine. More generally, it will bring softness, roundness, complexity, and participate to the bouquet of wine. But this is very dependent on the concentration it's present in wine. The usual concentration range from very small concentration, 0.05 milligrams per liter to 4 milligrams per liter. The perception threshold is matrix dependent, but usually, and you can see it here and it's been calculated in different matrices, but usually, and this is very general, it is considered an undesirable compound when it's over 5 milligrams per liter, and then it can bring over buttery notes, even going to rancid butter notes or even sweaty notes. Diacetyl has a very complex behavior in wine. Its presence depends on the conditions, the winemaking conditions, and also are dependent on the strain that is performing malolactic fermentation. On one side, we know that diacetyl, once it's produced in wine, can be enzymatically reduced to acetoin and then butanediol, which will reduce its organoleptic impact in wine. This happens during the fermentation and just after fermentation phases where you still have active cells, but this transformation is irreversible. On another side, diacetyl during aging can bind to SO2, and diacetyl that is bound to SO2 has a much lower organoleptic impact than free diacetyl. But you also have cell, or I should say strain dependent feature. First of all, you probably know that some strains, and this is a very rare feature, are incapable of getting citric acid into the cell. This transporter here that's called CYP doesn't work. So it's totally unable to produce diacetyl. Again, it's very rare, and on the market, there's only 1 or 2 strains that are marketed that way. After, among different strains, they have different ability to route a metabolite that is an intermediate to citric acid that is called alpha-acetolactic acid. Citric acid, maybe I haven't mentioned it, is indeed not directly transformed into diacetyl. It first goes through this intermediate, which is called alpha-acetolactic acid. And this alpha-acetolactic acid can either be transformed to diacetyl, and Interestingly, actually, this is a non-enzymatic reaction. It is due to leakage of alpha-acetolactic acid from the cell into the wine, or, and this happens in the cell, it is directly transformed into acetoin and then butanediol. And this equilibrium is strain dependent. There's also another feature that complexifies even more the diacetyl production in wine, is that strains have different abilities in the to degrade citric acid. They degrade it more or less rapidly, meaning that diacetyl will accumulate more or less simultaneously to malolactic fermentation. Let me show you a graph that will make that a little clearer for you, I hope. Depending on the strain, diacetyl will accumulate differently. Remember, once it's produced, it can be reduced, but it can be produced more or less rapidly because citric acid will be degraded more or less rapidly. You here have the examples of two strains, B7 and SB3. SB3 metabolizes citric acid very rapidly. So here's the amount of diacetyl at the end of alcoholic fermentation when SB3 hasn't had the opportunity to perform malolactic fermentation yet. And this is, in purple, the amount of diacetyl that is found in a wine that has been fermented malolactically with SB3 immediately after malolactic fermentation, 2, 7, 15, and 25 days after malolactic fermentation. And what you see is that for SB3, the maximum diacetyl concentration will occur very soon after malolactic fermentation, meaning that immediately after malolactic fermentation, diacetyl will start to be reduced. So the maximum concentration of diacetyl will be found soon after malolactic fermentation in a situation where SB3 is performing malolactic fermentation. The situation is fairly different with the B7 strain because it metabolizes very slowly citric acid. So you see that after malolactic fermentation, you still have to wait 15 days to reach the maximum concentration because during these 15 days, citric acid is still being degraded into diacetyl. This has a consequence if you want to stabilize your wine and use this stabilization procedure to optimize as a maximum or a minimum your diacetyl concentration. Let me be a little more clear. Here you have a graph with the two different strains, again B7 and SB3, and the diacetyl amount just after malolactic fermentation and 10 days after malolactic fermentation, whether this wine has been stabilized with SO2 or not. You see that for B7, if you stabilize with SO2 10 days after malolactic fermentation, you will have a lower diacetyl remaining in the wine, whereas if you haven't stabilized with SO2, after 10 days, you will have a high diacetyl, or a higher, I should say, diacetyl concentration because you will have not blocked the diacetyl the citric acid metabolism that again continues after malolactic fermentation. On the opposite, after malolactic fermentation, and immediately after you have I'd say optimized, a maximum diacetyl concentration that you will find if you have sulfited your wine after 10 days, and that you will not find if you have not sulfited your wine, because the absence of stabilization will enable the cells to reduce diacetyl during these 10 days. Okay, again, optimizing diacetyl concentration will depend on the strain you have and how far from the end of malolactic fermentation you stabilize your wine. Here is a table that will resume the situation. Let's look at it for the B7 strain. Again, citric acid metabolism is slow, so it's sequential to malolactic fermentation. Diacetyl production is moderate, but the maximum diacetyl level will occur late after malolactic fermentation, meaning that if you want a low diacetyl level in your wine, you will have to quickly sulfite your wine after malolactic fermentation. On the contrary, if you want a high diacetyl level, you will have to wait until the citric acid has been totally used, usually 10 days after malolactic fermentation. It is totally the contrary with FB3, but I haven't written this in this table because I must say that SB3 is a little less reliable in terms of managing diacetyl, and we have seen situations where to have a low diacetyl, you have to wait 10 days or sometimes 20 days, whereas B7 numbers are more clear-cut. Nevertheless, you see that depending on whether diacetyl is wanted or not, the microbial stabilization post-malolactic fermentation will not be managed the same way. And this is the main take-home message I wanted to give you. I'm done by with the Enococcus ani and looking at some of their substrate, malic acid metabolism and citric acid metabolism. I just wanted to finish to make a brief overview of different malolactic starters. Within the LAFFORT range, you probably know that you have 3 categories of products. Some are used for direct utilization, some need to be acclimatized before, and some need to go through a pied de cuve procedure to be able to perform malolactic fermentation. Let's look at the direct and pre-act products. In the direct product, you have 2 strains, B7 and SB3, that you can directly add to wine after having rehydrated them in 20 times their weight or volume of non-chlorinated water, and they can be used sequentially after alcoholic fermentation or as co-inoculation, whether early or late co-inoculation, meaning that you will inoculate them in must or in fermenting must. Another product that is called 450 Preac needs to be acclimatized before it's inoculated into wine, and it needs to go through a first step where it will be acclimatized during 12 to 15 hours in a mix of half wine and half water with a cell-derived product called Energizer that is a nutrient for bacteria. 12 to 16 hours of acclimatation are needed if you're in the case of a sequential inoculation after alcoholic fermentation, meaning that you're inoculated in wine or almost finished wine. But if you're in a situation where you're early co-inoculating, meaning maybe 48 hours after the addition of yeast, then a 30-minute acclimatation only is needed. There is another product that is called B16 standard. That is more a protocol based on a champagne type preparation that needs to go through a reactivation and then a pied de cuve step, either on must or on wine, that is more recommended for sparkling based wine and sometimes for malolactic fermentations in difficult musts. This product needs many different steps because you need to build a biomass before being able to use it in wine or must. This is the end of the presentation. It's not the end of the webinar. Please feel free to propose some questions during the Q&A session, and I think Kathleen is going to forward the questions to me now. Thank you very much for your attention, and again, if I haven't been clear on some issues, feel free to ask me for more precise explanation. I'll be more than happy to answer them. Thank you very much. Okay. I have one question. Can you please discuss the issues involved with co-inoculation of alcoholic and malolactic fermentation? Co-inoculation is a way to ensure a more malolactic fermentation. It consists in the natural adaptation of lactic acid bacteria because it will be inoculated during an alcoholic fermenting must by opposition to inoculating it in wine. So by inoculating it in the fermenting must, the cells will little by little adapt to a situation where you have increasing ethanol concentration and nutrition depletion. So it's a good way to secure malolactic fermentation, and it can be done either as an early co-inoculation, so as early as 48 hours after the addition of yeast, or as a late co-inoculation when the alcoholic fermentation has been through 2/3 of the sugar utilization. And in that case, let me precise that if you're using 450, again, the acclimatation time is not of 12 to 15 hours, but only 30 minutes. Kathleen, I don't know if we have other questions. Okay, well, thank you very much, and then feel free to, if you have reactions or questions later, to send them to Kathleen, and she'll forward them to me. Thank you very much for your attention, and it's time for me to leave you. Goodbye.